So this week we have Prof Daniel Anthony, the Professor of Experimental Neuropathology from the Department of Pharmacology, Oxford. And his research focuses on how inflammation contributes to the outcome of acute and chronic brain injury. So in his talk today, he will be highlighting the intrinsic differences that exist between the CNS inflammatory response and the peripheral response. So without further ado, please put your hands together for Prof Daniel.
Thank you. Thanks very much for coming tonight. It's a miserable night outside, so I totally understand. So I don't know whether, I'm not sure what all of your backgrounds are, so I'm going to be talking about something that we should care about because it's, the inflammation is now burgeoned in terms of the number of people that are sort of studying and caring about it, because I think we're establishing the fact that that the inflammatory response, certainly in terms of neurodegenerative disease, plays a contributory role. So very many years ago, it was noted by Pat Needy-McGeer that if you have Parkinson's disease or Alzheimer's disease, and you're taking non-steroidal anti-inflammatory drugs, and this is really what kicked off perhaps the beginning of the most interest in inflammation of the brain, that if you were taking these non-steroidals, you were somewhat protected against developing these diseases. So, of course, this generated a lot of interest. There was lots of studies were initiated, and the difficulty has been that it's never been absolute small studies seem to give rise to effects that can be observed, and it looks like non-steroidals might be good. And then as bigger studies come along, and then there were meta-analyses, and then sort of the impact of taking an anti-inflammatory sort of appeared to be less. But against that backdrop, because people became more and more interested in inflammation in the central nervous system, everybody started sort of measuring cytokines in the brain and realizing that actually their expression, de novo synthesis of cytokines, it seems to be associated with stress. It seems to be associated with a whole load of other sort of interesting neurocognitive disorders. In psychosis, we've discovered that there are different types of psychosis that are antibody-mediated, where seemingly antibodies are able to enter the brain from the periphery in a way that we never thought was possible. And so the whole sort of world of inflammation has grown somewhat. The reason it's entitled Misunderstanding Inflammation in the Brain is because we're going to have to divide this down. I'm going to talk about all elements this evening. Divide this down into inflammation that's associated with a pathology in the brain. For example, and I'm going to spend a bit of time talking about multiple sclerosis. Multiple sclerosis is an archetypal neuroinflammatory disease. If you look down the microscope, you'll see changes to the vasculature, you'll see recruitment of immune cells, you'll see plenty of activated microglial cells, activated astrocytes, T cells, hardly any B cells, but that's interesting because the newest therapies for multiple sclerosis are now, and the best perhaps so far, are B cell targeted. But there's CD4 and CD8 positive T cells, but there's an inflammatory response. You know, it's quite clearly a neuroinflammatory disease. We're also going to look at how other diseases, such as neurodegenerative diseases, prion disease, Alzheimer's disease, actually neurons die, and as a consequence of the death of those neurons, astrocytes and microglial cells become activated. And they become activated, and then the jury is out. So I told you about the cyclooxygenase studies, the non-steroidal anti-inflammatory drugs that are used seem to be protective. But fundamentally, it's not clear what those cells are really doing. Are they causal? Are they contributing to the pathology, or are they simply a consequence of having degenerating neurons? And we still remain a little bit jury's out. But I'm going to tell you how I think maybe the inflammatory response again has been misunderstood. And the way that they may contribute is in a punctate fashion, rather than being a sort of a fashion in which they're driving disease. But it's something that where if you have a peripheral infection or you have an environmental challenge of another sort, that then you can actually flick the inflammatory response from a prime to a sort of a rather aggressive response. And then, of course, we're going to talk about actually how if you have a peripheral systemic inflammatory response, so for example, I'm just getting over a bit of a cold. I have almost certainly have elevated CRP and SAA2 expression right now, and you know you've probably got a little bit of higher levels of circulating cytokines, and that changes the way that I want to behave. It means that you would perhaps rather be in bed. You don't want to see people. This is called sickness behaviour, and the study of sickness behaviour has spread over and into the study of major depressive disorder, because when you have a peripheral infection, the behaviours that you exhibit look awfully like the behaviours that you exhibit when you're suffering from depression. And that's, again, generated a whole new set of experiments and a field of research, neuroimmunomodulation. But what it's also done is it means that when you get de novo synthesis of a cytokine in the brain as a consequence of having a peripheral infection, actually what that really is it's modifying the way that the neurons are behaving, and it may not really be described as CNS inflammation. The problem being that microglial cells do sometimes become activated in response to a stress or an LPS challenge in the periphery. But we shall have a look at all that and see sort of where the direction of inflammation is going in the CNS. And it would be nice if my slides would move on. Let's just have a go at that. Great. Okay. The other thing that I want to talk briefly about is that usually it was just assumed that inflammation in the central nervous system is bad. So if you ever see microglial activation or astrocyte activation, then this is bad news. And I was down in Brighton recently, where John Maynard Keynes used to live with Vanessa Bell, part of the Bloomsbury set. And he wrote that the difficulty lies not in the new ideas, but escaping the old ones. And this is certainly something that's been very problematic for inflammation in the brain, where actually people see an activated microglial cell and they must always assume that it's doing something that's bad. Recently, with some friends of mine in Australia, we published a paper showing something that was really quite striking, actually, that if you completely deplete the brain of its microglial cells, the resident macrophages of the brain, so no more microglial cells, the animal's functionally just fine. And it makes you wonder, well, what is the point of a microglial cell? How can you remove all the microglial cells and you have no impact on behaviour? It seems pretty strange. It is a mouse, so maybe there are subtleties to the behaviour that you wouldn't necessarily see in a mouse. And then what we found was that it made very little difference to the outcome of a neural injury, of a TBI, traumatic brain injury. But where it did make a difference, interestingly, was in the recovery period, where recovery was improved if you had microglial cells repopulating. So if we allowed the microglial cells to come back into the brain after we'd injured it, then we discovered that these cells were now driving better brain plasticity than were they not there. So again, highlighting the microglial cell, when it becomes activated, what it might be contributing. It may not be there just to phagocytose and clear pathogens. It might actually be contributing to plasticity and brain plasticity and the formation of new functional connections, for example. Another thing that we know that microglial cells do is that they play a role just as astrocytes do in phagocytosing phagocytosing the wrong word, in taking up glutamate, for example. Astrocytes, we know, will clear glutamate. And when an astrocyte stops clearing glutamate, or sometimes pumps out glutamate, then it can contribute to neuronal cell death by an excitotoxic process. It transpires that microglial cells will put out processes and move them to high levels of glutamate in the brain, also seemingly responsible for taking up some of that glutamate. So once again, highlighting the fact that this response by glial cells, when they phenotypically become activated, they may not be doing what you think they're doing. So I'm going to just tell you a little bit. I've been in the game for quite a long time, so I, rather depressingly so, but it has enabled me to see sort of how things have changed. And it is interesting because the science, you might think, moves very, very quickly. In actual fact, it moves very slowly, but the sort of the fullness of time sort of stuff means that you can get some idea of what contributions that we have all made. So when I started, about 1983, which is a long time ago, these ideas were all completely well established. You know, there are no lymphatics in the brain. Absolutely. So basically, what's different about the brain is that nothing gets out of it. We would know at that time that leukocytes don't cross an intact blood-brain barrier. The brain is immune privileged. Also held to be true in 1993. For white blood cells to get across the blood-brain barrier, or to cross any barrier really, you need to have this expression of selectins and then integrins, and so it was said in 1993 that there was no selectin expression in the brain. It wasn't possible to upregulation of the brain endothelium, that there was no neutrophil recruitment in particular, that there was no B cell involvement in multiple sclerosis. I've already said that antibodies don't cross the blood-brain barrier, and that in multiple sclerosis in particular, that it's a demyelinating disease, and a failure to remyelinate the brain results in permanent disability. And it was also described that MS was one of those strange diseases that was sort of an incredible constellation of clinical signs, but it was still one disease. There was one therapy for MS. And at that time, when I was starting out, people were beginning to get interested in the connection between major depressive disorder and cytokine expression in the central nervous system. Now we skip forward. Nobody would argue that MS is just a demyelinating disease anymore. In fact, people sort of tried to pretend that that didn't ever really happen, and now we talk about MS as a neurodegenerative disease because we know that the permanent disability in MS is a consequence of the damage to axons and loss of neurons. We'll see some pictures. We know that axonal injury and neuronal loss are responsible for permanent disability, as I've said. There's this obsession now with lymphatics. Admittedly, it's rediscovered because some of the really early papers have noticed that lymphatics and the meninges did exist. There are lots of therapies for MS, but none of them stop progressive disease particularly. I mean, there are a couple that are now licensed for MS. We now know that, in fact, neutrophils, CD8, CD4, B cells can all cross an intact blood-brain barrier. And we know that all cytokines and adhesion molecules that we thought weren't expressed in the brain are expressed. And I mentioned the antibodies that we now know can cross. But what don't we know? We still don't know how it is that the brain is still so refractory, why the inflammatory response to the brain is so different from other parts of the body. And we still don't know about the connection between cytokines and major depressive disorder. And partly it's down to this sort of crazy nomenclature that everybody wants to talk about inflammation in the brain, but they're not willing to accept that actually these cytokines are being produced as neuromodulatory agents. They're not really an inflammatory response. It's not really about, there are no cardinal signs of inflammation, there's no increase in blood flow, and there's no leukocyte recruitment. So how did we get to this point? Let's just step back and just historically, Peter Medawar was responsible really for doing an incredible experiment in 1948, and he took an allograft and placed it into the central nervous system, and it doesn't reject. We can pretty much inject anything into the brain, and there's very little in the way of an inflammatory response against it. So I put a xenograft into the brain, and it survives quite happily. That was jumped forward 1994, the beginning of this work. It's hard to think that's such a long time ago, but transplantation of embryonic nigral tissue ameliorates functional deficiencies of Parkinson's disease, and that's by Anders Bjorklund and Olli Lindvall, highlighting again how the brain is a special place that you can put, and the individuals who have had that nigral tissue injected, when they failed to take their immunosuppression, again, the grafts didn't reject. And other examples of dendrocytes into the CNS of the Shiver mutant don't reject. So that's really how the notion of immune privilege came about, and that's why it was talked about so much. And then we had to try and explain why, and as I say, the why still isn't perfectly understood. But fundamentally, there are no resident dendritic cells in the brain. So that's certainly one thing that means that any antigen is going to have a hard time getting out. Potentially, depending on the nature of the antigen, it may be very soluble, it might end up in the spike of lymph nodes, but there aren't any dendritic cells to munch it up and then migrate and present. There are no resident mast cells in the brain. It's interesting. So, but you will all know that there's lots of histamine in the brain, so where does this come from? It comes from neurons. So that's, again, an interesting, we'll revisit that in a minute. And then, of course, it could be that it's all down to the fact that the microglial cells, these resident tissue macrophages in the brain, for which there was, for many, many decades, there was debate as to where microglial cells actually come from. But the idea was that the microglial cell was quiescent, and as a consequence couldn't do any additional damage, couldn't generate the cytokine response that would be required to enable leukocytes to be attracted into the CNS. But, having said all that, if you do generate a lesion in the central nervous system, so let's inject an excitotoxin, for example, into the brain, this is canic acid, into the hippocampus. And this is a guy that I worked with for many years, Hugh Perry, and what he noticed was what other people hadn't noticed, despite the fact they'd been injecting canic acid into the hippocampus to demolish hippocampal neurons for years, is he noticed that after you killed all the neurons, there was no acute inflammatory response. So normally, when you'd inject a truckload of an excitotoxin or any toxic element into any part of the body and it killed stuff, you'd immediately get neutrophils showing up, and you don't. Instead, all that happens is that the microglial cells become activated. They retract their processes, they round up, and this is really how we got, so basically a rounded up macrophage was one that was probably involved in some sort of process where it might even be contributing to the damage. But this was very typical, that you challenge the CNS, the microglial cells become activated, they round up, and then over time, day 35, they're basically indistinguishable again from the microglial cells that are there. So the idea of the microglial cells is quiescent and doesn't do anything that's wrong, but just looking morphologically at a cell isn't particularly helpful either. Just another example of how different the CNS is. So when you actually, if we take endothelin 1, which causes this time a stroke-like lesion in the brain, you inject it into the CNS, the vasculature shuts down, causes a sort of, generates what looks like a reperfusion injury in the brain. But what's striking is we kill new end neurons. We can see that compared to the ipsilateral hemisphere, that we lose neurons. There are no neutrophils recruited, although these are blue crescent-violet flame cells, but they're not neutrophils, they'd be brown. So no neutrophils. We get the activation and recruitment of some new monocytes, and that's quite an interesting thing. Is that there's a lot of debate about whether or not is a microglial cell really different from a monocyte, and it is true that they're differentially derived. Whether once a macrophage or a monocyte is recruited to the brain and becomes a macrophage, or whether it's an activated microglial cell, fundamentally they pretty much behave in the same fashion. I've never really come across an example where they don't. But you certainly get a response. But this is probably a response to the neuronal cell death, and it's probably a cleanup operation. Astrocytes become activated too, and as a consequence of the endothelin injection, you do get some damage to myelin. So the inflammatory response is different. You still get activation of the phagocytes, but it's microglial cells and macrophages rather than neutrophils. So, why is that? We now know that we can get these cells across an intact blood-brain barrier, and we also know we've sort of changed our mind a little bit about inflammation in the brain. What we now know, for example, is that when a microglial cell becomes activated and we switch on adhesion molecules, that cells can adhere to the vascular endothelium in the brain, and then they don't just punch their way through, they talk their way through. Both the cell becomes phosphotyrosine positive, the endothelium becomes phosphotyrosine positive, and we know that there's a rearrangement of the tight junctions. They literally part, let the cell through, and the cell moves into the brain, or the abluminal side of the vasculature. So all this can happen. But it's still clear that actually it's a relatively rare event that you'd ever get a neutrophil to enter the brain. But as I've said, something like multiple sclerosis is a neuroinflammatory disease. There is inflammation in the brain. So somehow in MS, this inherent refractory nature, where you don't get graft rejection, where you don't get neutrophils recruited, somehow in MS this is overcome, and we don't know why. That reinterpretation of privilege, therefore, means that, as I say, we know that cells can cross the intact blood-brain barrier. We know that tolerance to CNS antigens can be broken. This is kind of an interesting place. So if this goes back an awful long way, it's the French who did it first. They like injecting children with things. So Pasteur basically mixed up a whole load of he was making a rabies vaccine, but actually you grow the rabies vaccine in spinal tissue, and then mush it all up and then inject a child. And that child, obviously, develops a sort of a very strange paralysis. that looks just like multiple sclerosis. It's not actually, it's more like ADME. But as a consequence of seeing that the first model of multiple sclerosis was this child, and then subsequently we were taking myelobasic protein, MOG, and different CNS antigens. If you mix them up and you squirt them into the periphery in an adjuvant, the animal will develop this pathology, experimental allergic encephalomyelitis, EAE. And as a consequence of that, because the body has now become attuned and now recognizes the CNS antigen, it generates a cell-mediated response against that antigen by finding activated T cells find their way across the intact blood-brain barrier, and as a consequence, the lesions develop. So, if you break the tolerance to CNS antigens, if you expose yourself in the periphery to something that looks like a CNS antigen, then perhaps you'll end up with an immune-mediated pathology of the CNS. So with multiple sclerosis, in recent years, it's become clear that sort of our ideas that those proteins that must have got out, myelobasic protein, MOG, for example, against an oligodendrocyte, it's always been an assumption that those are the proteins that give rise to multiple sclerosis. As we know more and more, and I said that it was basically, MS was always thought to be one disease with a complex set of different symptoms. In actual fact, what we now know is that there are lots of different diseases embedded in multiple sclerosis, and a disease that we very much thought was MS transpires as an antibody-mediated disease against aquaporin 4, which is on astrocytes, and you get bystander tissue demyelination. So, and for multiple, and now a MOG-mediated disease as well, antibodies against MOG, and who knows what else will be teased off to find out whether or not there are other antigens that have got out. But perhaps this also explains how, for example, something like the limbic encephalitis develops, or indeed these antibody-mediated psychosis that I talked about at the start, that actually what we're going to discover is more and more elements of our behaviour or pathologies that we observe to do with targeting of different discrete entities in the CNS that we didn't think was possible before. I'm just going to highlight now why inflammation is such bad news in the CNS and when it happens, or when it's associated with a disease like MS. And also I'm going to highlight why some of the therapies for MS aren't very good. So multiple sclerosis, this archetypal neuroinflammatory disease, is associated initially with a relapsing remitting course. Almost, there is something called primary progressive, where people which may turn out to be a completely different disease, where people just get progressively more disabled as time passes. But let's start off just by thinking about what happens in a relapse in relapsing remitting MS. For whatever reason, an inflammatory response develops, an immune-mediated inflammatory response develops at the site of fairly random sites within the CNS. There is almost certainly some demyelination. As I've said, you get demyelination in neuromyelitis optica, where you have this antibodies against aquaporin 4. So we get demyelination and some remyelination. So that will give rise to some disability. You also get a lot of edema that puts pressure on neurons and stops them functioning, stops conduction. Inflammatory mediators, nitric oxide, for example, that will prevent or influence conduction. And because you get a little bit of demyelination, you might lose some synchronization. So that temporal and spatial summation that you know about will have disappeared. All these are recoverable, but what isn't recoverable is axonal injury and neuronal cell death, which is why anybody who has a relapse never really ever goes back to baseline. And what was shown by Hugh Mulder-Zeri and Barry Ferguson now a few years ago was that what they wanted to know was what was going on, when does that neuronal injury and axonal damage actually take place. It was always assumed that, as it started out in 1993, it was assumed that chronic demyelination of an axon would eventually cause the axon to give up, and that's where the disability arose. So it was much to do with a sort of a chronic phenomenon in the CNS. So what Barry and Mark and Hugh did is they decided to look at the relationship between axonal injury and the presence of macrophages. And what they discovered basically was that you only saw axonal injury in the acute lesions and in the border of active chronic lesions in the brain, and that these were associated with elevated numbers of macrophages locally. But fundamentally, what they didn't see is they didn't see any evidence of axonal injury either in the centre of a chronic lesion or in these sort of burnt out chronic lesions that existing throughout the neuroaxis. So what this told us for the first time was that axonal injury was a feature of, it was a primary event happening in an MS lesion. And in fact, this is what eventually gives rise to a brain that looks like this. And this isn't somebody with Alzheimer's disease, this is somebody with multiple sclerosis, secondary progressive MS. And you can see that the brain is looking really rather sad. The ventricles are massively enlarged and it looks nothing like a brain that you'd be used to. So this is really highlighting the fact that multiple sclerosis is actually a neurodegenerative disease, and that activation of microglial cells and macrophages in this instance gives rise to axonal injury at the time that you have the flare-up, this the relapse or an activated lesion in the brain, and that will summate eventually to give rise to a brain that looks a little bit like this. If you have multiple sclerosis, it's bad enough that you have the disability. Nobody wanted to know that they also have cognitive decline. It's now becoming as sort of as we've acknowledged the fact that myelin isn't the principal target, that it isn't about demyelination and failure to remyelinate. Actually, the bad news is that you lose your neurons. And in actual fact now, cognitive tests for MS are more common, and this is something where we need to go. So I'm very much highlighting here how the macrophage, the role of the macrophage in MS is associated with axonal injury. That activation here is potentially bad news, and that these macrophages are probably giving rise, they're probably expressing metalloproteinases that are demolishing axons of myelin, that they're releasing cytokines that's promoting the recruitment of other immune cells that can also contribute to the damage. So now what I want to do is, having said all that, I just want to illustrate a couple of things about these. So I want to backtrack again and now say the fact that you get an inflammatory lesion like that in MS is really quite striking on the basis of an experimental result like this. So here we've injected interleukin-1 beta into the brains of adult animals, 3-week-old animals, and aged 18-month-old animals, or TNF alpha. Same idea, we're going to inject it into the injection point is here, and then we look at blood-brain barrier breakdown in this particular instance. And if you put a bucket load of TNF alpha into the brain of an animal, it gives rise to no blood-brain barrier breakdown in the adult animal. And this is 3 micrograms of TNF alpha. It's a stupid amount of TNF alpha. That would make you really quite sick, and if you inject it into the skin, it would generate a foreign inflammatory response. Likewise, this is an injection of interleukin-1 beta, and there's a trivial amount of blood-brain barrier breakdown in the adult animal. What's striking, however, is there are periods during life, very early on, 3-week-old animal, where there's a window of susceptibility where we see a lot of breakdown with IL-1 beta, a little bit of breakdown with TNF, and you can see that actually if you then looked at the contribution of the inflammatory response, what sort of inflammatory response you can generate in an 18-month-old animal, you discover that the brain becomes susceptible again to both IL-1 and TNF. TNF in this particular instance, the brownness indicates a sort of a rather severe meningitis. Most of us do experiments on 8-week-old rats and mice. And so this means that if you're investigating the inflammatory response or the contribution of the inflammatory response, you're using an animal that probably isn't relevant to an individual who's going to have a stroke when they're 65, 70, 80. I hope it's going to be much longer than that for me. But highlighting here that actually the brain is remarkably refractory. So again, highlighting how surprising it is that people do end up with MS where that refractory nature is overcome. Right, so now just to sort of ask the question, so why is the brain so different? Why is it that graphs survive? Why is it so difficult in the normal brain to provoke an inflammatory response? And then it comes down to observations like this. This is by a friend of mine, Fernando Tosi and Vesadovsky, and they were able to show that that if you induce LTP in hippocampal slices, that you get an increase in the amount of interleukin-1 beta that's produced in those slices. Admittedly, it's a slice, which means it's a damaged piece of tissue. If we have a look at what happens then, if you think that IL-1 may be important, so let's put some IL-1 receptor antagonist onto the slices, and then we discover that blockade of IL-1 beta receptors in the slice basically demolishes the maintenance of LTP. So this means that a cytokine is influencing something that that a physiologist tends to just think about, this is about glutamate, and it's about how quickly the glutamate is released, and it's about the synapse, and why on earth should some IL-1 be involved? And then we look at the sort of the repertoire of other things in the brain that if you speak to an inflammation biologist about histamine, they'll talk about mast cells. If you talk to an inflammation biologist about serotonin, they'll talk about platelets. They'll talk about 5-HT receptors around blood vessels in the periphery. Prostaglandin, we just talk about the vasoactive effects of prostaglandin, how it's going to augment pain. And substance P, CGRP, that we now know, anti-CGRP, incredibly good for migraine. I promise you that's nothing to do with its inflammatory credentials here. And so what we see is that many of these mediators that we describe as inflammatory mediators in the periphery that are vasoactive, that encourage the recruitment of inflammatory cells, in the central nervous system they seem to be doing something rather different. We know that histamine does something rather different. I mean, anybody that's had to take antihistamines, if you take first generation or old-fashioned antihistamines, it makes you feel very drowsy. You feel drowsy because in the central nervous system, histamine is responsible for generating arousal. It's important. Serotonin, well, we all know that serotonin is all about happiness in the central nervous system, but in the periphery, it's really about shutting down those vessels and enabling platelets to aggregate. So it could be the case, and I put it to you, that basically the molecules that we're going to talk about, the cytokines, and in particular IL-1 tonight, the cytokines we're talking about that are made actually at relatively low levels in response to stress, in response to other challenges, are really just there as part of a normal immunomodulatory homeostatic system, and they're not driving inflammation. Let's just have a look at some of the things that we've done in the past, just for the sake of just to try. And over the years, we've really gone looking to see if we can find the reason why the brain is so refractory. So first of all, we think it's refractory because of the fact that you don't want to have an inflammatory response in the brain, because then it's going to mess up neuronal signaling. That was meant to be the take-home message from the last slide. That basically, if you generated a foreign inflammatory response, this is going to mess things up. If you have MS, you do suffer from fatigue. You do suffer from often from an atypical sensitivity to pain, for example. And so, it does seem that having those inflammatory lesions in the brain is perhaps altering cognition there, and your perception of peripheral stimuli. But let's see, as a consequence of those experiments we did with IL-1 and TNF-alpha, we were able to summarize a few things. We were able to show that common signaling pathways that exist in the periphery, the sort of the cytokine storm that people often talk about, which is really deeply irritating because it means absolutely nothing. But anyway, what we do know is that IL-1 in the periphery, IL-1 will induce TNF and TNF will induce IL-1. But in the brain, this simply doesn't happen. In response to a stimulus, and if we put IL-1 into the brain, it generates more IL-1, but it doesn't generate any TNF-alpha. And IL-1 eventually gives rise to the production of very specifically CXC chemokines that will give rise to a delayed and rather small recruitment of neutrophils after a delay to the CNS. Whereas TNF-alpha gives rise to the production of MCP1, CC chemokines, activates by astrocytes in particular, and then that gives rise to delayed monocyte recruitment. It's relevant because pathologies like cerebral malaria, for example, where it's strongly TNF induced actually gives rise to platelet aggregation and recruitment to monocytes, whereas there's no IL-1 that knocks around in that pathology. So we were able to show that actually it wasn't really about there being that these pathways didn't exist, it's just that they're highly specialized. We were able to show, as I mentioned, that the selectin expression, its E-anti-selectin, go up, whether it be adults or juveniles. So we were trying to look for the difference between what was going on in the adult versus those really young animals. So surely that could give us some insight into why the CNS inflammatory response is different, because if we could spot the difference between the adults and the juveniles, then we'd know. Sadly, just about everything we've ever gone looking for to try and explain that has never gone particularly well. When we look at, for example, in the adult and the juvenile, for the expression of chemokines, so that was an adhesion molecule, that's the chemokines, and there's really virtually no difference between the adult and the juvenile animals. So again, all the machinery from a pro-inflammatory challenge in the brain does seem to be switched on, but you still don't get rise to that sort of immune cell recruitment. So we thought, cleverly, and this starts sort of brings in the periphery and generates this sort of a new topic of study for us that began a few years ago. What we did notice, or what we did imagine might be happening, is that perhaps in order to get immune cells recruited to the brain, you need to generate an acute phase response, because you need to mobilize leukocytes into the circulation so they're available to go into the brain. So maybe there was no signaling pathway out of the central nervous system to the periphery to give rise to that mobilization. And bizarrely, what we discovered is that well before there was any kind of response in the brain to a CNS inflammatory challenge, well before, we saw activation of the acute phase response in the liver. And we saw an increase in the number of circulating leukocytes in the periphery. So neutrophils enter the blood just as they're supposed to, it's just they don't enter the brain. But we also noticed some rather strange things. We noticed that actually the neutrophils also seem to not only enter the blood, but also enter the liver as well. And this was so in response to a brain injury, the first thing you see is neutrophil recruitment to the liver. Whoops, it's gone the wrong way. So we obviously were quite intrigued by this and wondered how on earth this was happening. We were able to show that if we took these are some reporter mice where they basically cross a mouse with a firefly, and then you inject it with some luciferase, and wherever you have activation of NF-kappa B, which is basically upstream of the production of pro-inflammatory cytokines, wherever that happens, you get the production of photons. And so we inject interleukin-1 beta, microinjection to the brain, and the brain lights up with NF-kappa B. Makes perfect sense. But bizarrely, the liver, the whole of the liver, lights up even more so. And we found that was really rather quick. And a couple of other places found a little bit of the thymus, for example, but fundamentally it was the liver and brain that lit up with NF-kappa B. And then neutrophils were recruited to the liver. And that has ramifications, because after the brain injury, what we saw induction of hepatocellular markers of damage. So this means that you injure the brain and you injure the liver, and one of the features of a head injury is something called a systemic inflammatory response syndrome, or multi-organ dysfunction. And in actual fact, it looks like you injure the brain and then you injure peripheral organs. And this isn't making a huge amount of sense why the body would do that, except that any kind of brain injury is not something that you would, from the evolutionary perspective, you'd normally get away with. Anyhow, so those neutrophils that enter the liver are bad news. And still left the question, how does this work? And now, to cut a very long story short, we looked for every humoral mediator we could think of. We thought there'd be perhaps IL-6, maybe it was just IL-1 getting out of the brain, maybe it was TNF, maybe it was something else, but it certainly had to be something in the circulation. And we discovered that, in fact, it was none of these things. What it turned out to be were these small blebs that come off, in this case, astrocytes in the brain. They're called extracellular vesicles. They bleb from the astrocyte, they cross the blood-brain barrier, and they target the liver, where they switch on hepatic cytokine production, and that gives rise to the mobilization of neutrophils and goes back to the brain. Let me explain the graphs that I'm showing you here in the experiment. So, altemusine is a inhibitor of neutral swing myelinase, and it prevents external vesicles from being produced. So there will be no increase in external vesicles. We then inject that animal with interleukin 1 beta into the brain, but it can't generate external vesicles. We still get adhesion molecule upregulation, so selectins are expressed in the brain, whether it just be IL-1 alone or IL-1 beta with the neutral swing myelinase inhibitor. But then what we decided to do was we isolated the exosomes, external vesicles, from this animal and injected them into the animal that had been treated with the neutral swing myelinase inhibitor. And we discovered that actually we could reconstitute the animal, reconstitute the inflammatory response, and neutrophils were now recruited to the brain again. So effectively what this is saying is I injure the brain, I switch on external vesicle production, they enter the circulation, they target the liver, they switch on hepatic chemokine production, where that gives rise to the mobilization of neutrophils to the bone marrow that are then free to enter the brain. And you have to have all. Why is this interesting? This is interesting because getting drugs into the brain is really tricky. If you want to target or prevent immune cells from being recruited to the brain, this now gives you a whole load more targets to actually choose in terms of going after that. So, where do those extracellular vesicles go? Well, they seem to go to Kupffer cells in particular. And if we deplete Kupffer cells from the liver, we discover that we can reduce the number of neutrophils that are recruited to the brain. So we go from this to that in the absence of having any liver Kupffer cells. So before these experiments were done, nobody would ever believe for a second that a Kupffer cell in the liver was responsible for controlling the magnitude of brain injury. That was still a very, very reductionist idea. IL-1 beta into the brain, and so let's actually injure an animal this time and find out whether or not the same sort of things apply. So we did a contusion injury in the spinal cord and found that in the presence or absence of Kupffer cells in the liver, we could reduce the number of neutrophils that were recruited to the injured spinal cord, or the number of ED1 positive macrophages that were recruited to the injured spinal cord. So there's the potential here to suppress the inflammatory response in an injury model and do that in such a way that you could prevent that sort of secondary inflammatory mediated damage in the spinal cord, or indeed perhaps in the brain. These are some experiments we did where we basically took plasma EEVs from an injured animal, or from macrophage EVs that we elicited by treating them with, I think it was TNF-alpha in this instance, or some where we stimulated endothelial cells, again I think it was with TNF-alpha, to generate extracellular vesicles. And in each case we injected them into an animal with a TBI, and increasing the number of circulating EVs increased the severity of the lesion. So highlighting the fact that, so we've got signals that leave the brain, go out in order to initiate an inflammatory response, initiate the mobilization of leukocytes so they can go back in where they could cause further bystander tissue destruction. The way in which now we should just have a look at the flip side of that. So let's now imagine that you have a peripheral inflammatory response and we have signals coming out. We're sending leukocytes back to the injured brain. But now what I want to do is we're going to actually think about what happens if you have, say, a lung injury or some other challenge or even a stressor, and how, for example, activating the acute phase response or having peripheral circulating cytokines might influence the way that the brain is now behaving. So we're going to go in a slightly reverse direction. And the interest in this has spiraled out of control because of experiments like this, where if we now look at the plasma TNF-alpha levels in bipolar or major depressive disorder, we discover that for IL-1 or for TNF in the circulation, having higher levels of cytokine in the periphery seems to be associated with major depressive disorder or bipolar. So that's really sort of, now we're talking about, let's go the other way. So those peripheral cytokines, wherever they came from, higher in the periphery seem to be in some way interacting or perhaps maybe are driving these neuropsychiatric disorders. So in our own hands, the way that we can model that is we can perhaps stress an animal and then look at the cytokine levels in the CNS. And we've discovered that although in an inbred set of animals, there are some animals that are resilient and some animals that are susceptible to a chronic mild stress. By the way, the chronic mild stress isn't mild at all, really, and we can't, I must say, we take the tissue from other people who do this, where they get ethical approval, etc., but I certainly wouldn't try and get sort of a license to do chronic mild stress in Oxford. Anyway, when you do that, the stress-susceptible animals, we see that there's an increase in the number of IgA1 positive microglial cells in their prefrontal cortex, and we see that in the susceptible animals there's an increase in the expression of TNF-alpha and an increase in the expression of interleukin-1 beta. So, stress is inducing cytokines in the central nervous system. And that stress and those cytokines seem to be giving rise to this sickness behavior, where cytokine expression in the brain can give rise to, in animals, the depression of mood, a modified sensitivity to pain, depression of motor activity, can induce some fever, anorexia, a deep depression of exploratory behaviours, and altered sleep behaviour. Interestingly, those are sickness behaviours that are common between rodents and humans. We do exactly the same things when we've been challenged, when we've had a viral infection, when the viral infection has been cleared, and yet we still are left with this sort of horrendous malaise that exists. One thinks of long COVID in this instance, perhaps. But also, you know, basically if you've had chikungunya or dengue or EBV, frankly, the same sort of things. You know, I think it's going to be interesting to see whether long COVID is actually any different from any other sort of post-viral response. How is that mediated? The way that we think it's normally mediated is sort of an allostatic homeostatic response. So you have a systemic inflammation, you switch on the hypothalamus, the NTS by the vagus, we then switch on the pituitary to activate the HPA axis, and you make a bunch of cortisol and some noradrenaline, and somehow this keeps everything sort of balanced. So if you have a systemic inflammatory response, in order to feel okay and to make sure the inflammatory response doesn't go out of control, that you end up with this balanced system. Clearly, sometimes, if the systemic inflammatory response is excessive, it's possible that you will end up with an inappropriate activation of the HPA axis, and in this case, we can end up with sort of, as I say, inflammation running out of control in the periphery. Likewise, we can imagine that we have brain injury, as I've been talking about, where you get these, where you actually get sickness behavior from having a CNS inflammatory response. In this instance, then we also end up with an atypical or an aberrant immune response in the periphery. So, for example, after a spinal injury or after a head injury, you suppress, the spleen actually shrinks after the injury. So there's a bidirectional system that controls behavior and manages to sort of keep everything homeostatic if possible. This sort of work, as I say, has gone back to 1995. And the way that we now mimic in particular, so this sort of cytokine-induced depression, is by cheating a little bit. So instead of using chronic mild stress, we just take some lipopolysaccharide and inject it into an animal. That's a bacterial cell wall protein, bacterial cell wall product. And what we're going to do here is look to see, this is a milligram per kilogram LPS actually, so this is pretty high. And what this does is that if we look at the way of assessing whether an animal is depressed is by giving a sucrose preference. So what we do is we look, in this case it's saccharin, but sucrose is acceptable too, and we discover that if we give an animal some LPS, then it doesn't really care whether it drinks sweetened water or non-sweetened water. So this drops off to about 50%. These are almost perfect results, just to let you know. So it's which is nice, browse is great, but we often have a hard time getting a perfect result of that. So there's no preference, it's 50-50. What is remarkable is that with pre-dosing and giving a mipramine plus the LPS, the animal now is happy to drink the saccharin, prefers to drink the saccharin. So effectively we're using an antidepressant, a tricyclic antidepressant, and we're showing that that is able to reverse the LPS response. Nancy Rothwell and Manchester did it with SSRIs and IL-1. Lots of other people have shown that basically you can reverse these behaviours with typical antidepressants. So again, this is sort of pushing forward this idea that basically the cytokines are in some way driving depression. I'm going to argue and put it to you that this is not really depression, this is malaise, and there are certain things, certain behaviours that synergize, and some that are additive, and some that are actually negative. I mean, this is some work that we've done. It just highlights how when you take that LPS challenge, the time of mobile and the forced swim test goes up. The time that the animals are active in the open field, the distance travelled is reduced. Time they become anxious as well because they reduce the time in the central zone. And we can see that the LPS, not surprisingly, if you inject an animal with LPS, you would get an induction of an acute phase response, so this is SAE2 in the liver. But what is quite striking is that we have an increase in TNF-alpha in the hippocampus, and in the prefrontal cortex, which we tend to focus on quite a lot, we have an increase in IL-1-beta and TNF-alpha, so this elevation. In recent times, not surprisingly, we've been using TLR7 agonists for looking at sickness behaviour. This is rosiquimod, and showing that actually we can induce very similar sorts of behaviours. They do have sort of rather nuanced differences between mimicking a viral challenge and using a bacterial cell wall product. We tried using a compound that we thought was going to be a cure for COVID, and sadly doesn't do an awful lot. You can see there's very little difference between the Fanostat here and our vehicle, which was a bit depressing. But anyway, it enabled us sort of to build and understand this model of sickness behaviour. To a greater or lesser extent, I'm afraid that many of these models, simply what people are reporting on is the fact that glucose basically dumps out of your blood. And it's taken us, as I say, about 30 years to get to this point where we're sort of acknowledging that actually an LPS challenge makes you feel shit because you have no glucose in your blood. I think all of you will appreciate that glucose is a fabulous drug. If you've ever had a child that gets irascible, or you get irascible, and you need to have some sugar, then you feel better. And to some extent, this is exactly what's happening with LPS. Now, there are elements of behaviour that persist for an extended period after the glucose returns to normal. So that actually is perhaps sort of mimicking that sort of this post-viral ME-like syndrome more than looking at the acute phase. But I promise you, most of the papers that have ever been written using LPS have examined the models when glucose has been very, very low in the blood of these individuals. Right, what about mixing infection with stress? And here we found that we used 0.5 milligrams per kg. We don't like a big per kg, and we think that kills a few animals actually. It's not really very good. And you can see that it makes them 0.5 mg per kg. Does all the things we'd expect. This is the elevated OMAs, a test for anxiety, and we see that latency is increased as a consequence of 24 hours compared to the vehicle. Going back down at 48 hours, very little persists at 48 hours. And we see that we get total number of exits is reduced by this amount. This is a resident intruder test, and sort of interactions are reduced by this dose. You'll notice that 0.1 mg per kg does virtually nothing. So let's take that dose, because we know that dose still induces changes in the expression of serotonergic receptors in the brain. So it's still having an impact, but not so that we can measure in our behavioural tests. And now let's have a look and see what happens when we combine stress, that chronic mild stress, with an LPS challenge. And effectively, mostly, things synergize. So the sucrose preference test, you put that additional, that bit of LPS that normally doesn't do very much. So we're basically talking about a sort of a subclinical challenge with LPS. So you're not really feeling that much worse, although it must be said that rodents can tolerate an awful lot more LPS than we can. But anyway, we get synergy. The sucrose preference test, we get exacerbation of that. Exacerbation, sucrose intake actually goes down. On the tail suspension test, the duration of mobility goes up. The duration of floating in the forced swim test goes up. So across the board, we have synergy between a stress and a low-dose inflammatory systemic challenge. But other things also happen that don't fit quite so well. For example, just do that again. So what we see, hang on a minute, was it that one? Let's go back. Yes, let's have a little look at other bits and pieces that are happening here. So if we take stress and LPS, stress alone increases in the resident intruder test. We get an increase in the number of attacks. But so the animals become aggressive when they're stressed. We give LPS and that aggression goes away. The duration of interaction as well, as you can see here, in terms of is ameliorated by the stress plus the LPS. So it's not always synergizing. Sometimes it's sending behaviours in a slightly different way. And then we look at cytokine expression, and we discover that in actual fact, actually, and this is a really important slide. It doesn't look like an important slide, but it's quite an important slide. Because what it's highlighting to you here is if we just look at the expression of IL-1 beta, for example, induced in the raphi, or just about anywhere for that matter. So let's look at TNF alpha as perhaps a better example. We look at the induction by stress, and then we look at the induction when we bung in some LPS, and we discover that actually there is a considerable difference in terms of that endogenous. But LPS is generating a lot more cytokine than stress alone. It always comes down to this amount. Mostly, stress induces cytokines, but not a lot. And if I injected LPS into the brain, I promise you this would be orders of magnitude greater as a consequence. Anyhow, we're seeing that what happens is stress plus LPS, and we get an amelioration. LPS alone this much and falls off, and that's sort of chewish across the board as well with other things. So sometimes with the expression of cytokines, they don't actually go in the way that you expect. The way we expect some synergy there, that if stress induces IL-1 and LPS induces IL-1, we'd expect to see more, for example. So, quite different stories. In terms of what goes on in the periphery, and that liver story that I told you about, we see that if we give some LPS, we get hepatic TNF production, which is fine, and then bizarrely, if we add the stress, this is ameliorated. What is striking in so many of our experiments is that basically cortisol never really accounts for the changes that we see. In terms of sort of stress-resilient animals, cortisol stays the same, and you can see here that actually LPS, it wasn't just the case that we had managed to massively ramp up the production of circulating corticosteroid, and that somehow gave rise to this reduction in hepatic TNF. It's basically corticosteroid independent. Cortisol in humans, corticosteroid in us. This is an interesting one about how the LPS actually works, and just a very brief aside really. We put LPS into animals, and we're often wondering how the LPS actually gets to where it needs to go. And we did these sort of quite interesting experiments where we mixed LPS with HDL or with LDL, and we found that effectively, LDL, we all know LDL makes you feel better, and it does indeed. So we discovered that if we inject LPS, we get an increase in IL-1 beta, for example, in the prefrontal cortex, and if we mix LPS with LDL, that's reduced. LDL alone, there's no change compared to the saline. And then we look at the HDL, and we put in LPS, and if you have LDL that's HDL that's mixed with LPS, then you get an increase in cytokine expression in the prefrontal cortex. So this is showing an interaction between your circulating lipoproteins and the LPS challenge. Again, quite an interesting idea about, we think that lipoproteins are far more important in terms of signaling than anybody gives them credit for. It's not just about moving cholesterol and fat about the body. Good. So just to summarize what we've got to then, chronic stress and immune stress, basically aggression, hyperactivity, impulsivity, and helplessness, all signs of depressive-like disorders in humans, and reported to be present in animal models. But when you combine a low dose of LPS and chronic stress, you get an enhanced depressive-like phenotype, but significantly reduced manifestations of aggression and hyperactivity. So, highlighting the fact that we can't just assume that sickness behavior is the same as depression. You stress an animal, one set of behaviors. You induce sickness behavior, it's a different set of behaviors, and they don't synergize in the way that you'd necessarily expect. What if you have, so that's without a CNS pathology. In the last few slides, I just want to show you a few things relating to the relating to what happens with peripheral systemic challenges if you have CNS pathology on board. And to start, we're just going to go back to multiple sclerosis briefly. Animal models of multiple sclerosis, but it is well known. So Sibley many years ago showed that Bolger actually were able to show that, systemic inflammatory responses, UTIs, respiratory tract infections, seem to precipitate relapse in multiple sclerosis, but mechanistically nobody really knows how that happens. And there's this mismatch between the challenge and the relapse. The challenge usually seems to precede the relapse, but and we'll see why that is in a moment. But this is just highlighting, it's a model we use, the model, we generate a focal inflammatory lesion in the brain. It's associated with all the sort of the MRI changes you'd expect in humans. There's gadolinium enhancement. There's an increase in the signal on a T2 weighted scan, and revealing the fact that you get local blood-brain barrier breakdown, and then it resolves. And what happens, which is really interesting, is that the axonal injury is ongoing. So although by MRI you cannot see the lesions anymore, the lesion is actually still there and it is still causing damage. So that's why radiology can be a little bit misleading in the case of CNS pathology, and especially in the context of MS. In order to see that ongoing, so you'd have to kill the animal and look for axonal injury. We can see inflammation, and we can see that because we can use this is a PET. This is called GE180. It's a radio tracer that binds to activated microglial cells, and this is highlighting the fact that if I give a drug called saponemod or FGY720, figolimod, sorry, or which is an S1P modulator, from 21 days from day 127. So the beauty of this model is we can take it out an awfully long way, but this is a time when you cannot see any pathology in the brain, but we know that microglial cells are still activated in these lesions. So what happens if you have a systemic inflammatory challenge? So this is plus or minus LPS. If we plus LPS and we load the animal up with some iron-containing or labeled macrophages, so we give a bunch of macrophages some little iron oxide particles to eat, and then inject them back into the peripheral circulation, we discover that a peripheral challenge with LPS causes new leukocyte recruitment to the brain at those totally invisible quiescent lesions, seemingly quiescent lesions, and an increase in axonal injury. So highlighting that you have to be worried about what happens in the periphery if somebody has a CNS disease. This is also the case, or another example, we've done this in a number of different animal models. This is the first one we've made. This is a model, this is prion disease. And this is the normal appearing microglial cell. This is a microglial cell in prion disease, heading towards the end stage. And you can see they've all activated. And this is a stain for interleukin-1 beta. And you can see that actually in this animal, with all those activated microglial cells, there's basically no IL-1 beta being produced. But if you give a systemic inflammatory challenge, an LPS challenge to these animals, the cells look the same, but in actual fact now what happens is that they start producing an awful lot of IL-1 beta. They have become primed, and then they can contribute towards neuronal cell death. And those animals will die 2 weeks earlier than they should. So, what we're highlighting here is that in the context that inflammation in the brain, when you have a systemic, when you've been stressed, or you have an infection, generates cytokines in the CNS, where it modifies your behavior. It's part of that, probably part of that homeostatic response to make you be able to cope with what's going on, reprioritizing your behaviors so that you can deal with that injury or infection. But not inflammation per se, it's actually a physiological response. Then we see And what happens is sometimes you do have an inflammatory response in the brain, truly inflammation. The vasculature changes, you have leukocyte recruitment. And sometimes that inflammation can be rather diffuse, sometimes it can actually be MRI invisible. But if it's there and you have a peripheral challenge, then what happens is those microglial cells can become activated and start releasing free radicals and other cytokines where they may give rise or contribute to further neurodegeneration, inflammation, and toxicity. But normally, for the most part, those microglial cells are probably just engaged in brain plasticity and homeostatic functions and will have an impact on cognition, but not necessarily in that sort of the way that you think. So, in terms of what we do therapeutically, maybe the thing to do is to think about, we're not going to give an anti-inflammatory because inflammation is contributing to all these processes. Maybe what we need to think about is, it's a normal process. Actually, immunomodulators might be a more sensible, well, not even immunomodulators. We need to think about what cytokines are doing in the brain in response to stress, in major depressive disorder, in psychosis, and other things where we've got cytokine expression in the CNS, and maybe anti-inflammatory isn't the way to go there. So in those situations where we have CNS pathology, or we have a neurodegenerative disease and you have systemic inflammation, we need to time the therapy. So we suppress, we give antibiotics more freely and faster, we give antivirals more freely and faster if somebody has a neurodegenerative disease, to try and sort of nip that interaction that we know exists between the periphery and the CNS. And so I think that's really why just calling something inflammation isn't helpful. So I'm having a little look at where we've come from and where we might be going to. I don't know why I chose 2044, but anyway. Well, except of course that I will have retired, which is very nice. I haven't talked about it. We do a lot of work on the microbiome. I'm such a skeptic, but people keep coming to me and saying, I want to work on the microbiome. You say, oh, God, all right, you can work on the microbiome if you must. And so we do lots of microbiome work. And oh my God, don't we just do fecal transplants? I mean, it's just gross. But it's in animals, just to make it clear. It's still slightly less gross, but anyway. And you can just gavage. You don't have to do it that way. Quite a lot. Anyway, so real-time microbiome monitoring will be possible by clone drones, colon drones. You know, I genuinely think this is something like this is going to happen well before 2044, I'm afraid. MS will now be many different diseases. I think we've only started peeling off the segments here. Progressive MS hopefully will be achieved by probably selective depletion of the population of CD8 positive T cells. Right now it's obsessed with B cells, but I think it was initially CD4 positive T cells. I think that CD8 positive T cells are the only ones. We'll play with the lipidome. We'll be modifying our circulating lipoproteins. But let's cut to the chase. I think we will eventually understand that cytokines and chemokines are simply neuromodulators and part of normal physiology. And I think that the cytokine link between cytokine expression and depression sadly will still remain completely opaque for most people and for us. But anyway, I'm going to end there. Thanks very much for your time tonight.